workshop.
Tracy, the way you're presenting,
Yes.
You have a square that's projecting, and it's probably something there. It's gone. All right, great.
Great. Everything okay now? If so, then welcome to our workshop this morning. Just like yesterday, let's see. Let me see if I can advance the slides. Very good. Just like yesterday, we have a few requests for the group to try to make this WebEx format work as well as possible for everyone. So everyone has been muted upon entry, and we're going to ask you to please stay muted and to keep your video off throughout the workshop to preserve as much bandwidth as we can for the presenters. But please do ask questions. Please use the chat feature to do that. And today we'll ask you to send them to everyone so that we can address as many as possible. If you have any technical issues, please contact our AV support team, and their contact information is down there at the bottom of the slide. So today, what we're going to try to do is to help you integrate biomarkers more effectively into your early phase trials. Biomarkers are more and more an essential part of drug development in oncology, but biomarkers have their own development timelines and requirements, and usually those don't mesh comfortably at all with drug development timelines. If you're starting from scratch, it can take 2 years or more to develop a sophisticated, fit-for-purpose biomarker assay, whereas you want to get an early phase trial up and running inside of a year. So how do you make that work? Usually, your best bet will be to recruit a collaborator who has already done a lot of the development work on the biomarker that you're interested in. But how do you know what kind of a lab to look for? What kind of questions will you need to ask them, and what questions can you expect from them? And why does it take so long? So what we're going to do today, our plan is for me to make a few initial remarks, get you started with a list of questions, and then we're going to provide you with presentations from 4 labs that are examples of the kinds of collaborators that are going to be best equipped to help you. So each presentation is going to illustrate what goes into making a biomarker assay fit for purpose on a different platform. Each presentation will illustrate a different platform. They will give you a glimpse into the development process that's already taken place before you ever come calling. They will discuss the kind of biomarker effect that platform is best equipped to measure. And they will spend a lot of time on the pre-analytic requirements for the specimens that are required for that kind of an approach. So then, armed with that background, we are going to ask you to put your trial developers' hats on and to tackle a challenge that is based on an actual CTAP project team. We're going to go into some breakout sessions for that. Each one is going to be moderated by one of our lab experts. We'll only have about half an hour to do that, but we'll try it. And then afterwards, Dr. Jeff Moscow, who is chief of the investigational drug branch at CTAP, is going to pull us all back together and wrap things up. So to begin, during trial development, there is a stage of brainstorming. In early phase trials, there are so many interesting and unanswered scientific questions that could be addressed as correlatives. And those go up in a whiteboard, and they serve as the beginning of the conversation with the lab. But that conversation has to refine that list. It has to boil it down to a much more limited and realistic selection of biomarkers that actually get written into the LOI. So what we're going to try to do today is we're going to show you this trial team's basic concept and what their whiteboard looked like in the challenge. And we'll briefly model that conversation because it really is the key to the effective biomarker plan. But how do you get started? How do you get ready for it? The first thing to recognize is that there is no linear checklist for moving through the process. Every decision that you make about the design of the trial and the first biomarker that goes into it is going to constrain what follows. And through the course of your conversation with the lab, you're going to encounter a number of reality checks. And we're going to illustrate some of them as we go through the morning today. And what you need to do is to confront them early as you can, and make your decisions accordingly. You'll be starting with a concept for the trial, an initial concept, and this is going to impose an initial set of constraints on the number and the type and timing of specimens that you can expect to collect. And this is going to be your first reality check. You won't be able to do everything. You don't want the biomarker plan to become a problem for the trial, delaying activation, messing up accrual. You need to focus. So one way to start looking for laboratory partners is to ask for your trial, for your particular trial, whether it's more important to focus on a pharmacodynamic question or a predictive question. Is the drug working? What patients might be expected to benefit from it? If you go the pharmacodynamic route, here's some of the first questions that you can expect your pharmacodynamic laboratory collaborator to ask you. What kind of samples can you collect? What would be the right time points? How big of a drug effect can you expect on the analyte that you've chosen to measure as an indicator, which might or might not be the target of the agent? Hopefully you have answers to some of these questions from prior preclinical work or from prior trials. If you really are at square one, it might be better to recognize that early and plan for that accordingly. But hopefully you can find a collaborator who's already done the work to develop an assay for the biomarker that you're interested in. And if so, the conversation can proceed in more depth with your collaborator. A lot of the same ground: effect size, sampling strategies, time points. You're going to have to work through all of these questions, these that are up on the slide, and the ones that follow from them, as our laboratory experts are going to illustrate in a moment. But if, on the other hand, your focus is on a predictive biomarker, but there's no reliable test that would be readily available to your patients, then you'll be looking for a collaborator to set up a laboratory-developed test for your trial. And these are some of the first questions that collaborator is going to have for you. Prevalence is obviously critical. And for starters, your prevalence estimates you have in the literature, but they are almost always going to be based on a wide variety of tests, and the confidence intervals are going to be very wide. But this here is the really hard part. Most biomarker data is continuous, and it needs some kind of cut point or threshold in order to make decisions. But in order to have any confidence about your cut point, you're going to have data from some patients who were enrolled on the trial, even though they might not be expected to respond. And it gets harder and harder to do this as drug development proceeds. It's sometimes a very difficult decision to know when to start selecting your patients on the basis of a biomarker test, but it's critical for the development of an eventual diagnostic based on the biomarker, just as it's critical for the drug. So this is a hard question that is going to need time for a full discussion with both the trial team and the lab. Either way, you need to understand CLIA. If the test result is going to be reported back to the patient, such as for an eligibility test, that test will need to be done in a CLIA certified lab. And if it's a brand new lab developed test, that CLIA certified lab is going to need time to build it and to validate it for your trial. If the test result is correlative and it's not going to be part of the medical record, then the lab does not need to be CLIA certified. You will note from the presentations that you're about to hear that Dr. Pavlovich's lab is CLIA certified, whereas Dr. Ferrigalo's is not. It doesn't have to be, but it's very CLIA-like in many of its procedures, as she will describe, and that's what's actually important. So as you go seeking collaborators, expecting these questions, I think, is going to help you recruit the right kind of lab to work with. They should be asking you these questions, and if they're not, you might be in the wrong place. So at this point, because finding the right collaborators is so important, what we've done is to ask several people who direct those kinds of labs to come today and show you how they go about building assays that are fit for purpose for early phase trials. I'm going to start introducing them now. So please do start using the chat function to send questions their way. And I believe the first of the speakers on our agenda is Dr. Stephen Hewitt, who is head of the Experimental Pathology Laboratory here at NCI. And let me figure out how to stop sharing. There we go.
Good morning. 1 second as we make the magic of virtual happen. My computer is thinking about this, of course. Is the screen showing a large Navajo weaving?
Yes.
Then we have succeeded. Okay. Good morning. I'm Stephen Hewitt. And 1 sec, I got to move something up. Yeah, I'll live. I'm going to be talking about some of the pitfalls of immunistic chemical assays in clinical trials. I'm a translational pathologist at the NCI and play roles across a broad spectrum of this area. Okay, come on. Oh, there we go. Let's try this button. Okay, there we go. I've been trained as a Fed. I always a good boy and tell you some background of me. I belong to the government, and it's a warning that I edit a journal. So I think all of you probably know what a biomarker is. However, people have a tendency to not understand that a biomarker really is a decision point. They don't embody it. And, it is something that you are going to have to live by the consequences of, especially if you've built it in as a predictive biomarker into a study. And so you have to embody this entire concept as you're going through testing. Today, we have large numbers of testing panels, and you get lots of extraneous data. And when you get a result that is, quote, extraneous, but is a decision maker, you have to change your thinking. And it's always something to keep in mind. So I'm going to be speaking about immunistic chemistry, and this really remains the front line of pathology diagnostics in the biomarker field. Histomorphology is still a critical tool, and we use to categorize tumors, but increasingly we're using immunistic chemistry to evaluate prognosis and predictive biomarkers. Molecular provides a substantial amount of information. In fact, I practice molecular pathology, but the fact is that immunohistochemistry is cheap, it's easy, it's fast. It is available across a broad spectrum of facilities, and as a result is highly desirable for your biomarkers simply from an implementation perspective. And that's one of the major drivers in which it's such a popular biomarker today. It's familiar to pathologists, it's familiar to clinicians, and increasingly it's actually familiar to patients. Molecular biomarkers do cause patients a lot of quandary. So I use this pyramid. It's important to understand where this system is and what's going on, because oftentimes when you're looking at an immunistic chemical biomarker in a trial setting, you're talking to your trialist pathologist who is going to interpret your test. And that's wonderful. You need to have a strong relationship with this person and have a broad conversation with them because they're really doing a lot of work for you. However, oftentimes that frontline pathologist, that sign-out pathologist, is not the person who's going to be developing the test or validating the test. You have a lab director and his staff who run the test and perform the quality assurance. They may, in some instances, be the test developer, but oftentimes they are not. The test developers live in a translational pathology world, and they develop and validate the base test. And this is what happens at our own institution. Oftentimes, I'm working with investigators who come to me, and they have an idea for a biomarker. They want to investigate a gene of interest. And I build an immunistic chemical assay for them, and I carry out a diversity of activities in developing and understanding that test. And then when it's time for it to move into a clinical trial, I hand it over to the director of our immunohistochemical laboratory, who will perform another level of validation, assure that the proposed scoring matrix is effective, and then we'll find and hand it off to a trial pathologist who's going to interpret it. And so these are a very complex process. And so just going to the pathologist and say, can we do this test, is not going to get you where you want to go oftentimes. You need to be talking to the guys who actually build these animals and validate these animals and run them in clinical settings. And we'll be talking about some of those issues. Diagnosis with a capital D. I love to play language games with the word diagnosis because it means different things to different people. And I oftentimes wrap all threes, diagnosis, prognosis, and prediction of response, into the D word, diagnosis. But really, diagnosis with a small d is the classification of disease based on histomorphology and cytomorphology, and is what my bread and butter job is. Prognosis is prediction of outcome. Constellation approaches and tools are available and has much more qualitative interpretation. Whereas prediction of response biomarkers are calibrated and quantitative and statistically validated. And they're going to have clear, clean, and performance understood performance about the cut point, which we'll talk about again in a moment, hopefully. Immunohistochemical assays actually follow the same structure. The majority of biomarkers that we use in immunohistochemistry are markers of lineage. They're supportive but not definitive. A classic example is S100. Not all melanomas are S100. You can have an S100 negative melanoma. You have to interpret the immunohistochemical stain within the context of the data that's available to you. They are nearly always scored positive or negative, the presence or absence of an individual protein. And as a result, their assays are actually formulated in a slightly different manner so that they give you exactly that answer. I'm positive or I'm negative. And you will hear pathologists use unusual and mystic language like scattered, few, and rare. And those actually mean specific things, hopefully in a regular language to a pathologist. Prognosis, associated with an outcome. Doesn't tell you the outcome of that individual patient, but tells you the outcome of a large number of patients. Again, most frequently scored as positive or negative, but they can be qualified as, strongly positive, weakly positive, or you can even have a system such as the all-red system. The challenge is that most prognostic biomarkers are still moved over into a world of positive or negative. There's a cut point, and if you're above that cut point, you have one outcome, and below that cut point, you have another outcome. One of the challenges with prognostic biomarkers is combining them. You have two biomarkers with each binary cut points. Most of the time, you've got four potential outcomes. You add more than that, you can see the multiplicity of outcomes that bedevil you. And so this is a chronic issue for many investigators and pathologists. Predictive biomarkers are going to guide therapy. And as I've said, they should be calibrated and they should be quantitative. And they should have performance standards and an understanding of performance about the cut point. One of the challenges about this is the opportunity to modify that performance about the cut point. One can put an indeterminate section in there, which many people don't like, but really has the benefit of modifying certainty functions with reference to the calibrator and the outcomes. And so there are different ways that a predictive biomarker can be deployed. We're demanding more of our test. As I said, immunohistochemical assays were really developed for lineage, and their scoring was rather subjective, and we've moved to a very objective world. Pattern is obviously very subjective, and pathologists are good at it, and they can even tell you an enormous amount of prediction of outcome and prognosis just on the subjective evaluation of histomorphology. But as I said, the drive is toward objective. We've also moved from descriptive, the florid language of dermatopathology, to a pathology that is more qualitative, enumerative, and quantitative. And it's always a challenge of how we're going to use this information. As Tracy noted, we're talking about quantitative data and clinical decision-making, which is binary. And one of the challenges is the validations and the regulatory approaches that the FDA uses to address these issues. It obviously gives you statistical power to help you, but you have to learn how to work your way through the system. Okay. Lab-developed test, LDTs, assay calibration, and predictive biomarkers. LDTs are also colloquially known as homebrew. And to be honest, we've seen an enormous amount of discussion about LDTs and homebrew lately with COVID testing, because many of the laboratories that are providing it are actually building and validating their own assay. And it requires an enormous amount of skill and a large number of samples to effectively do this. One of the biggest problems that we have encountered across the entire field of oncology is efforts to build LDT predictive assays, where they claim that the results are equal to the calibrated FDA-approved assay. And the challenge is oftentimes they use statistically insufficient samples to determine if this is correct or not, and don't have a calibration that looks at the outcome of the patient. A true predictive assay was built against a data set for which the performance of the outcome of the patients is determined. And this is very challenging. Non-inferiority test is not required. It's not really the best way to deal with this. The FDA continues to struggle with this, but it's something that is going to continue to be a major issue across the development of predictive biomarkers. Conceptualization of a test. This is something that most investigators really don't need to embody or worry about, but the pathologist does. And it is something that should come up in a conversation. And that is, what is the intended use of the assay? How are you using it? Again, as we've talked about, what's its utility, but also what's the contextual application of its use? Is it being used at the diagnostic, or is it being used at some later time point? Are you performing the test on a sample that was acquired at the original diagnosis after the patient has received therapy? These are all very important questions that need to occur. Sampling and time periods are unusually complex. Validation of performance is another issue. On the other side, we're talking about total test. This is the construction of the assay itself. This is a concept, again, that's probably unfamiliar to most of you, and really is unfamiliar to many pathologists, but should not be. And it's a modular approach that allows us to modify a test through its life cycle and not have to just completely start over. And it involves the pre-analytic, which hopefully I will talk about in a moment, but the analytic and the interpretive. The pre-analytic is the specimen, and the specimen includes the patient. Analytic is the assay itself, and the interpretive is the interpretation of the results of the post-analytic. And we'll be talking about the interpretation later. As I mentioned, there's the life cycle element of it, and that is an assay is always going to change over time. The ER assays we use today are not the same ER assays that we used 20 years ago, and we have to keep this in mind. And it's very easy to start believing that, well, I have a better reagent, so it inevitably makes my test better, and that is not necessarily the case. And validation of specificity, are you measuring what you claim you're measuring, is very critical to all immunistic chemical assays. Validation of these assays, we separated into molecular validation and clinical validation. The molecular is the binding of the target antigen, and it is dependent upon the antigen specificity, the controls, and the defined or definable sensitivity and specificity of the assay. The clinical validation is performance within the intended use. And this is where investigators get into trouble. For example, new antibodies for ER came out a few years ago that were very, very sensitive. And there was this belief that they would make estrogen testing better in clinical samples. The problem is that the ability to find ER in lymphocytes in the tonsil of a woman really has nothing to do with the response of her breast cancer to estrogen therapy. And it was really the clinical performance that needed to be evaluated, not the assay performance in and of itself. Okay, again, as I mentioned, total test, we have the pre-analytic. You have the patient, the endogenous factors that are going on. Pathophysiology is very complex. People have a tendency to think that a biomarker in a patient is stable. This is far from true. For those of us who live in studies of the kidney or even COVID, you see a substantial oscillation of a biomarker, and you're never certain, is it pathophysiology or is it normal physiology? A classic example in the oncology world is estrogen receptor, one I keep talking about, and the fact that groups have tried to use normal breast epithelium as a calibrator for ER expression, failing to appreciate that normal physiologic ER expression is a variable that is going to vary against menarche, a woman who is cycling, and menopause, and that even a woman who is cycling will have monthly variations. And so ER is not an effective endogenous calibrator. There are exogenous factors, which are the handling and storage of the material. And these are the ones that bedevil my life. And I actually spend a great deal of time both trying to understand and evaluate for our biomarkers. Two great examples of problems with biomarkers in this space, in fact, are HER2 and PD-L1, both of which notably lose their performance with unstained slides over a period of anywhere between 6 weeks and 3 months. And this has a high likelihood of giving you a false negative result. This is even more complex in a world of both immuno-oncology as well as long-term survivors, where we oftentimes have a tendency to evaluate the patient's original samples. What you'll do, oftentimes I'll be asked to test a sample that is between 5 and 10 years old. And at the immunistic chemical level, if it's a block, it will probably perform okay and return a true and accurate result, although there are a couple of biomarkers it may not. On the other hand, if one is evaluating RNA, at the age of about 5 years, a paraffin block is starting to really lose its quality in RNA and may not provide appropriate results. Analytic performance, the reagents, and the performance of the assay. This also includes the training of the staff and the hardware, maintenance, and can even include the environment that the hardware is stored in. And we have seen that multiplex immunistic chemical assays, in fact, can have some challenges where cryptic software updates will change the results. And these are things that one has to worry about when you're a test developer. Interpretive phase. The good and the bad news is over the last decade, we have seen the challenges in clinical trials move literally down the system. Previously, it was pre-analytic variables that were bedeviling us, and then we had a small phase of analytic variables that were bedeviling us. Now it's become interpretive phase. And that is the interpretive paradigm reproducible, and is the quantitation effective and good? And definition of interpretation of results. And these are the new boundaries that we're facing today. Pain points in immunistic chemical assays. I think I kind of covered this in a moment ago, but archival versus trial-based samples is a big challenge, and sample adequacy is increasingly a big challenge, especially in the world of immuno-oncology, with patients receiving needle biopsies to evaluate their current disease rather than evaluating their original excisional material that might be 5 or 10 years of age. As I mentioned, we made a lot of progress on analytic performance, and a great deal of this can be attributed to high-quality automation. People ask me in the field of immunistic chemistry, are stainers good? And the answer is flat yes. There's never an example where automation really isn't better than trying to do this by hand. And in interpretive, we have these challenges which I've discussed, and one of the issues is the disconnect between the results and the application within the clinical trial. And that is, you have a biomarker that is well validated and has excellent performance, but its interpretation really does not fit within the metrics being desired for the clinical trial. One is looking for, say, a percent change of a biomarker that is largely non-quantitative but is binary. You're then suddenly expecting that 50% of your cells are going to shut off your biomarker. And some of those examples just don't bear fruit, and it's become a challenge for us. Reproducibility. This has become a big issue through NIH granting mechanisms. It's something I believe in a great deal, focus on reproducibility and validation. And this is one of the paradigm examples we use about trying to understand our reproducibility. It's built on the controls and the validation, but it's also built on the adequacy of the samples, the sampling approach, and the calibration. It's not difficult to find yourself in an instance where the sampling alone is inadequate and adds to the noise of the biomarker because you're just looking at too little tissue. When you're looking at tumor cells, you should have some threshold of the number of cells you're evaluating. And a great example, again, is estrogen receptor. If you've got a cytology sample and it's got 10 tumor cells and one of them is positive, and you've got a cutoff of 10% for positivity. Oh, you've won. You're good. Everything's great. On the other hand, if none are positive, but you only have 10, how certain are you that sample is truly estrogen receptor negative? And that's a tumor example. Move that over to the field of immuno-oncology, where you're trying to evaluate the presence of lymphocytes within a tumor, and it becomes even more challenging. And calibration of assays is a big issue. Biologic calibrators are very tenuous and difficult to use. Exogenous and truly standard curve-based immunistic chemical assays are exceedingly rare. I've published and worked on a couple of them. They're fantastic and they're wonderful, but most of the time they're actually dealing with exogenous controls that are artificially built and provide a true standard curve, which is not generally available on most immunistic chemical assays. A simple metric that one should consider immediately is performing the assay in your lab with your samples, collaborator specimens in your lab, send your specimens to his lab, and then cross them back over. This really helps you address all of those issues of reagent type, specimen type, reagents, hardware, methods, and interpretation. And I'm not going to tell you that this will clean up every assay, but it removes probably 75% of headaches in the early phases of clinical trials. Just because it's CLIA does not mean it's fit for purpose. CLIA is very focused on the assay, not necessarily on the test, because its focus is on reproducibility, controls, reagents, and protocols. It doesn't include the concepts of intended use, the setting of use, doesn't really address dynamic range, and it certainly doesn't address the interpretive dilemmas of sampling, scoring, and reproducibility of interpretation. Context of interpretation. This is very challenging. An assay can have multiple contexts. Estrogen receptor is another good example I keep coming back to, and that is in the breast, we'll use it to determine if the tumor is ER positive, ER negative. But in a patient who's 5 years out and presents with a positive lymph node or a lesion in the lung, we're just using ER to help us define that, in fact, the breast cancer, that it's an ER positive tumor and narrow down the possibility of tumor types. Sampling adequacy, I think we've talked about, and a lot of it's driven by biologic heterogeneity. And performance about the cut point is really a challenge, trying to be certain that you have true false positive, you limit the number of false positives and false negatives. How can you refine a test? Well, you can use cell lines. Those have limitations, but they can be powerful tools in the early testing phase of building an assay. You can use xenografts. Again, they pose certain problems, especially if you're in the immuno-oncology field, but they move you to another level and ask additional questions. One of the advantages of xenograph systems is they help address heterogeneity much better. Model organisms, when appropriately chosen, are a powerful tool, and of course, archival samples are never to be diminished in their importance. More is always better, and nothing ever replicates performing an immunohistochemical assay in a real setting. I may take a biomarker and run it through retrospective samples extensively, but until we push it into a trial, we never really understand its performance. First in human, this is a big topic. You really need to build in a biospecimen plan early. And one of the challenges that we often face is the desire to build a predictive biomarker early in the clinical trials, where you really cannot define the final aspects of the predictive biomarker until later stages. You may be developing and refining that biomarker in phase 2, but you're not really testing it better until phase 3 or later. You need to have strict control of your specimens increasingly. Biospecimen depletion has become a major problem and is limiting many, many trials across many platforms. You need specimens from untreated patients for baseline, always an important issue. Assay development on the fly is something you want to try and minimize as much as you can. Frequently, you'll find somebody who's moving through their trials and suddenly discovers they need a biomarker. Well, if you didn't have those biospecimens planned coming out from the beginning of the trials, you're usually behind the 8 ball. One of the biggest challenges we face is the disconnect between the drug target and biomarkers. Oftentimes it's assumed the biomarker is the drug target is the best biomarker. That is oftentimes not true. You need to be a little bit more agnostic. And there can be some scientific gaps between mechanism and metabolism of drug, and I've run into those previously. Application of existing tests without re-engineering for intended use was an example where you have a deployed biomarker, an immunohistochemical assay that's used in a diagnostic sense, and that you want to try and move it into a prognostic or predictive value. And again, sometimes you can overcome these problems, but it may require recalibrating the assay entirely. Some laboratories will in fact offer a particular biomarker with different conditions for immunohistochemical staining to meet these needs. It's not unusual to find a number of different of the same target with multiple biomarker assays in the laboratory, and you have to know which one needs to be deployed. And this is a discussion to be had with a trialist pathologist. I think I've covered most of these. I've talked about the normal tissue conundrums. Another one that's critical is assay reproducibility, day, week, and month concepts. We've run into headaches with this. You'll run into, especially you need to demonstrate that it is reproducible and stable with reference to the specimens and the reagents and the platforms, and all the way to the technologist. A strong reproducibility schedule is do it on Monday, do it on Friday, do it the next week with a different tech, and look at what you get, or do the next month with a different tech and compare those results. And you need to actually do a sufficient number of samples to be certain that this is what you're hoping for. Laboratory to laboratory is another critical element. As some of these technologies become more advanced, you discover that just minor differences in either settings of the instruments or how the staff use them will impact the results. And so harmonization and calibration is critical across some of the more exotic multiplex platforms. We've talked about scoring systems. Cell type and tumor type, illogical use of the same scoring system. An example is the efforts to try and use the original HER2 scoring system for breast on gastric cancer. Didn't quite work. There are other examples as well. Another fine example is trying to use a scoring system for HER2 for HER3. And well, related molecules, and they form heterodimers. Why can't I use the same scoring system? And the answer is you can't. I routinely get investigators who approach me and ask me to tell them what the scoring system is for a marker I've never stained, and I just look at them and say, no. I also will say that the magical histoscore system, which everybody desires, really doesn't perform as the way you think it does. It just looks really cool on paper, and that other scoring systems may give you equal or better information and capacity to interpret data. Manual scoring. To be honest, manual scoring cannot be beat, because if it manually scores well, it's going to be robust. You do need to look at inter and intra-pathologist performance. But if you have a biomarker that performs well in inter-pathologist performance in a manually scored system, you have a much stronger biomarker than a biomarker that's going to require sophisticated imaging and image analysis, which may be much more subject to pre-analytic and analytic variables. And so it really should never be diminished that the eyes of a pathologist sometimes are your best friend. Pre-analytic drift is something that can be a real challenge. One challenge element of this has been the smaller tumors at presentation, but also differences in handling and degradation, which are hard to evaluate. Analytic drift, I've already mentioned about, quote, improvements in reagents, but changes in hardware also have to be evaluated. A CLIA lab will actually have to validate every new instrument. And then I've already talked about clinical versus assay sensitivity and specificity. I actually run a lab, got a lot of staff. Without them, I can't talk about these fancy subjects. My email address at the NIH. I'm always happy to answer questions. I do a fair amount of consultative biomarker practice. And that's it. Thank you.
Thanks very much. We have one question in the chat. Thank you for someone putting it up there. You mentioned that automation is nearly always, but you mentioned actually that manual testing is nearly always superior. Let me get this backwards. You mentioned that automation is nearly always superior to manual testing, but what are some common pitfalls to avoid when migrating a historically manual assay into an automated process?
That's an excellent question. And the biggest pitfall that we run into from going from manual to automation is actually oftentimes the changes in reagents required for the assay. Many people will have an excellent manual assay. I run many of my assays semi-automated, and our antigen retrieval will be performed offline and our deparaffinization will be performed offline. I use a very stringent deparaffinization, and we use some pretty rigorous and rugged antigen retrieval approaches. And when we try to take those to our clinical instruments, what we discover is that we have to switch antigen retrieval systems. We're going to switch the deparaffinization programs, and sometimes we're going to switch the detection system, but we're going to maintain the primary analyte reagent, the antibody, the same. And so you're going to have to retiter the antibody, and you actually do need to compare the two straight forward. So that happens to be probably the most common bug that we run into is the necessary change in reagents.
Great, thank you so much. I think it's time now to move to the second speaker in our series, which is going to be Dr. Kate Ferrigalo from our Clinical Pharmacodynamics program up at NCI Frederick.
So it would help if I unmuted myself. Good morning. Great, let me pull up my presentation. Here it is. Okay. Okay, is that, does everyone see that okay?
Looks good.
Great, thank you. Good morning. This presentation, Clinical Implementation of a Pharmacodynamic Immunofluorescence Assay, will be delivered this morning by me, by Kate Ferrigalo, and Dr. Ralph Parchman, my co-presenter, is going to pick up towards the latter part of the talk. I'm a principal scientist, and Ralph is the Senior Managing Director of the Clinical Pharmacodynamics Biomarker Program at the Frederick National Laboratory. And we're happy to have this opportunity to speak with you this morning. Okay, I'm having, oh, here we go. Let me do something else real quick. Pointer. There we go. Okay. So before I get into the main portion of the talk, what I'd like to do this morning, and to Tracy's important comment that really maybe one of the more important things to know when faced with or when you have the desire to implement a study, a pharmacodynamic study, as part of an early phase clinical trial, is really knowing who to collaborate with, what your resources are. So I'd like to take just a few minutes to update you on the pharmacodynamic assays offered through the National Clinical Laboratory Network. And then the main portion of the talk are really highlighting those important principles, those important factors to consider when clinically implementing a pharmacodynamic assay, and those features which are important for implementation of a high-quality assay. We are using quantitative immunofluorescence assay this morning as an example. However, really, any high-quality pharmacodynamic assay should consider and incorporate these important principles. I think by now, most of you are familiar with the National Clinical Laboratory Network. Just briefly, currently this kind of body of resources is at the heart of it are 3 laboratories. The NCLN Genomic Laboratory, which is harmonized by assays developed and validated and utilized by the Molecular Characterization Lab, also at the Frederick National Laboratory, and you will hear from Dr. Chris Karlovich in the next presentation, who helps run this program. And there are 2 pharmacodynamic assay laboratories. One has been established at MD Anderson for quantitative immunofluorescence assays, and another at a laboratory in Tennessee, Molecular Pathology Laboratory Network, for the implementation of pharmacodynamic assays using extraction-based sandwich immunoassays on the Luminex platform. The National Clinical Laboratory Network also provides infrastructure for these studies, including access to a centralized biorepository to help manage the specimen shipments and storage, make sure the right samples go to the right laboratories, and also access to centralized data analysis tools. I provided some links here for more information about the National Clinical Laboratory Network, as well as some of our own within the PD program resources that we have made available for our validated clinical pharmacodynamic assays to the clinical research community. This is sort of a busy slide, but I do want to highlight some important points here. What I have here listed are assays that have been developed and clinically implemented or at various stages of clinical implementation by the PD program in Frederick. The ones that are shaded in red are already available or will be available soon, have been approved for movement to the National Clinical Laboratory Network. Currently, we have, there's 2 bullets, one for the quantitative immunofluorescence assays and the other for the Luminex assays for the sandwich or for the extraction-based assays. The assay currently available in our PD lab at MD Anderson is the DDR3. So we are accepting applications through the protocol approval process for access to this currently, and we have several others at various stages of clinical implementation and use within our program that will be prioritized for transfer to that laboratory in the future. And knowing, again, commenting on the importance of collaboration. Obviously, collaboration with the right group of people is not just access to a tool, but hopefully access to a body of knowledge, the scientific knowledge behind the targets and the drug actions that led to that particular measurement rationale. So with that thought, I've also tried to provide several important references about these methods. So currently DDR3 is available, and we will prioritize others in the future. For the Luminex assays, we currently have an intrinsic apoptosis multiplex assay available to be applied for that resource. And we do have approval, and we're in the process of beginning to move several panels that are, this is the intrinsic apoptosis is currently available. And the PI3 kinase RAS-RAF signaling multiplexes, which are constituted by 5 panels, we have just begun to take the steps to begin transfer those methods to the NCLN laboratory to make those available. So getting into the main portion of the talk, there's a very important distinction to understand between analytical validation and fitness for purpose. We know that analytical validation is important for any assay measurement, sensitivity, specificity, dynamic range, et cetera. And that's very important for establishing the suitability of your measurement for the intended use of your measurement. But in order to really establish suitability of an assay that's intended to support an early phase clinical trial, you must also establish fitness for purpose to show that that is ready for that clinical situation. Fitness for purpose studies are performed in our program in a model system and using a complete model system that simulates the clinical trial as closely as possible. For example, using the most relevant preclinical models available, a relevant therapeutic regimen, and a relevant tumor sampling procedure. You see here a picture of a mouse. This is a live mouse being biopsied with an 18-gauge needle and rapidly flash frozen. This is exactly what we would do. Obviously, it's not exact to what's going on in a clinical setting, but we try to replicate that as much as you can reasonably do so. And the main purpose of these fitness for purpose studies is to demonstrate a significant drug effect, given all the known, the obvious biological and technical variability that's really inevitable for all types of measurements of this nature. But it's, again, that's what's shown here, to be able to establish in this setting a very significant drug effect, and very importantly, to establish a time frame for that significant effect. Important to know, will the clinical trial logistics allow for tumor sampling during this relevant time frame? So once you know that time frame. Demonstrating a pharmacodynamic effect using a baseline. We had comments from Dr. Hewitt about baseline. Knowing the baseline variability of a biomarker is relevant in relevant specimens is important using the intended assay methods, and that helps to establish whether or not you're likely, or you're in a situation where you're very likely to establish a significant effect at the drug dosages planned. So knowing the baseline is important. And then also can help you establish which type of clinical trial you could use your assay in, or that it would be suited for. For example, in a phase 0 trial, the dosages would be non-toxic. And so depending on if you have low or high baseline variability, the response curve is very relevant for that in order to determine if that measurement is suitable in that situation. For quantitative immunofluorescence assays, understanding measurement variability across a biopsy is also important in understanding what the minimum cell count is to reach significance. So once you have a certain number of patients that you've gotten a response in a drug-treated specimen, knowing what that minimal cell count is very helpful, and then understanding if this is achievable with most of the specimens that you are receiving. Biopsy suitability is a topic really dear to my heart. We've spent a lot of time really thinking about this with the pharmacodynamic assays over the last several years. And it's now recognized clearly as a significant challenge for successful biomarker studies. It's very important to understand that there's a big distinction between a sufficient research biopsy and a fit for purpose or sufficient diagnostic biopsy. Archival tissue is not a substitute for a pretreatment biopsy. And in our program, we have found that flash freezing of biopsies within 2 minutes of collection is really critical for success. So we've implemented an SOP and in our own program, and we've trained others to have individuals ready at the point of collection to obtain that specimen immediately and flash freeze that. And very important for labile biomarkers, and many important pharmacodynamic biomarkers such as phosphoproteins are in that category. Improvements in improving biopsy suitability, and we've definitely achieved that over the years. There's a number of factors involved. Probably most importantly is very robust and regular communication between oncology, radiology, pathology, and the PD laboratory, not only on the requirements of the specimen up front, what do you need and what your measurement is, but also in communicating back the success rate. If you're seeing a lot of failures, getting that information back to the oncologist and the radiologist quite quickly. Use of additional cores per time point, use of improvements in biopsy fixation and processing, and other factors such as use of tissue segmentation that I'm going to touch on a little bit further now. Tissue segmentation for accurate PD biomarker assessment. This is basically in any, and Dr. Hewitt also commented on this, is that cellular composition or heterogeneity is sort of an accepted complicating factor in adequacy and the analyzability of research biopsies. You're very typically having a very heterogeneous, sometimes low tumor content biopsy. And just quickly use it, this is just showing you the use of beta-catenin as a tissue segmentation marker in a quantitative multiplex immunofluorescence assay to highlight these tumor areas in this low tumor content biopsy and be able to highlight those in the region of interest in a microscopy image. If you were just looking at the image without the segmentation, you would falsely perhaps enumerate all of these nuclei in the field, whereas with the segmentation marker, you're able to pull out nuclei only in the tumor positive areas, and that leads to a very high, greatly increased accuracy of your pharmacodynamic assessment. Again, this salvages poor quality, I don't want to say poor quality, low tumor content specimens, very heterogeneous specimens, which are very often seen. We use beta-catenin in our program it works well for most carcinoma and some sarcoma, and we are working on developing additional tissue segmentation markers for particularly for the sarcoma category. Benefits of multiplexing. Multiplexing on any platform for clinical trial analyses preserves valuable specimens by maximizing mechanistic information per slide or per specimen. It provides those additional channels for segmentation or phenotypic markers. It also improves the likelihood of finding a signal in the sampling window. And in an ideal situation, could provide corroborating information. If you see one or more additional biomarkers responding, it gives you more confidence. In this case, on the left-hand side, this was a patient that was a partial responder in a drug trial with a lead-in of irinotecan and then a second biopsy with the addition of veliparib. And we saw no increase in gamma H2AX, the more traditional biomarker for DNA damage response in this particular patient. But with the addition of phospho-MBS1 in this multiplex, a very robust increase in phospho-MBS1 was evident in this particular patient. Assay quality control. This is something that is very important. Tracy mentioned CLIA-like, and we have a lot of metrics in place to not only develop a robust fit-for-purpose assay, but also to maintain quality of those assays through the sometimes very long accrual time and utilization of assay to support a clinical trial. Stringent incoming testing for reagents is absolutely essential, and it will bury you very quickly if you don't have qualified reagents for your assay. Also, the use of controls to monitor run-to-run performance is essential, as well as SOPs to really lock down all methods from the very beginning, from the point of biopsy collection to the reporting of the clinical data, as well as rigorous methods for operator and laboratory certification and proficiency evaluations. So thank you. And with that, I'm going to turn it over for a few additional comments from Dr. Ralph Parchman.
Thanks, Kate. Good morning, everyone. So in the last couple of slides, I'd like to just cover some practical aspects of conducting clinical PD studies. On this slide, I want to address the issue that in some of the trials, a baseline biopsy is not available. And for a pharmacodynamic study, this raises the important question of how a molecular drug response could be demonstrated in the absence of a baseline comparator. And if it can be demonstrated, what should the on-treatment biopsy be compared to? So in this situation, there will be recommendations to substitute the patient's original diagnostic biopsy in place of a current baseline biopsy as the PD comparator. But in addition to possible differences between the original and current disease that you heard Dr. Hewitt refer to, there are also those inherent procedural and pre-analytical control differences between diagnostic biopsies and PD biomarker biopsies that Dr. Ferrigallo alluded to just a minute ago. And this is especially apparent, these differences between diagnostic and pharmacodynamic biopsies are especially apparent when the biomarker for the BT study is a labile biomarker, such as a post-translational modification of protein. For example, protein phosphorylation in our hands, for example, in the MET receptor, has shown a degradation half-life of about 3 minutes, even under cold ischemia conditions. So that provided the rationale for, in the previous slide, you showed Kate present the standard procedure for us, which is to snap freeze the specimen within 2 minutes. That's to preserve even the most labile phosphorylation residues that we've studied so far. So there's also an emerging body of knowledge on this subject of differences between diagnostic biopsies and pharmacodynamic biopsies. And this slide lists several NCI publications that address the topic in detail, and also describe important aspects of procedures, biopsy procedures, and specimen handling, both in the clinic to the lab and then in the lab itself as the specimens are thawed out for the analysis. So I'd refer you to those references for more information. Next slide. Okay. So then a related question is there some type of study design that can be incorporated into an early clinical trial that would allow for the assessment of a molecular drug response even without a current baseline biopsy comparator? And it's possible, and I've listed some of these possible designs on the slide for consideration. I would point out, at least for us, and I think generally in the field, there's not a lot of clinical experience in using these designs to demonstrate a drug response in the absence of a current baseline biopsy. But I would point out toward the bottom that where it says randomize initial biomarker patients to different time points, that this has been tried even in the clinic with an NCI phase 0 trial of the PARP inhibitor veliparib, where the patients at a given dose level were randomized to the on-treatment biopsy a few hours after the dose versus 24 hours of the next day after the preceding dose. And that allows one to demonstrate a time-dependent change in the biomarker value. And if the fit-for-purpose modeling has established that that is an expected result, that goes a long way in improving a molecular drug response in the patients, despite the lack of a baseline biopsy. So at the bottom, we have some important questions to consider when thinking about pharmacodynamic studies and incorporating their designs into your early clinical trials. And especially, what are the pros and cons of risk of waiting to use a fit-for-purpose validated assay until it's critical? So, for example, waiting to measure the drug response until MTD or the expansion cohort is underway, versus using dose escalation to establish that the timing of the on-treatment biopsy provides evidence of drug response, that there's actually measured response. And so you know that you're on the right time point when you go into your critical cohort of patients. And then finally, there's the question of whether pharmacodynamics relates to tumor response measured by clinical measurements like RECIST, radiologic imaging, or outcomes. And of course, critical to that in pharmacology is that we're able to measure the maximum effect of the drug on the target or the downstream consequences of that target modulation. And with a single time point, just keep in mind, it's very difficult to know whether that signal is the Emax, so the maximum effect, or whether that signal is rising or falling. And so without knowing what the maximum molecular effect is, it may be more difficult to try to relate it to outcomes and to do PD/PK modeling. So next slide, I just wanted to thank everyone who's involved. This, of course, can't happen without a integrated team of clinicians and laboratory staff and experts in various aspects of clinical assays. So I just want to thank all of the collaborators on this team and the patients who have participated. Thank you very much.
Thanks very much, both Kate and Ralph. I hope everyone was paying attention during those slides that Kate was showing about the importance of allowing enough tissue for the success of the pharmacodynamic assays. I don't see any questions in the chat window at the moment. I think if people would like to enter questions in the chat window for Kate and Ralph, they'd probably be able to respond asynchronously. But if there are no questions at the moment, in order to stay on time, I'd like to move ahead at the moment with our next presentation, which is going to be by Dr. Chris Karlovich from the Molecular Characterization Genomics Laboratory up at NCI Frederick. Chris?
All right, thank you for that, Tracy. Start my video. Good morning, everybody. I'm going to pull up my slides right now. Just give me a moment here. All right. Tracy, can you see those?
They're visible.
Fantastic. All right. Let me go to projection mode. All right. Well, good morning again, everybody. So my name is Chris Karlovich. I'm with the Molecular Characterization Laboratory. We call ourselves MOCA, and we are also, like Kate and Ralph's laboratory, we're also at the Frederick National Lab for Cancer Research in Frederick, Maryland. Mickey Williams is our director. I'll be giving the presentation this morning. I'm the associate director, and our mission at MOCA is to provide genomic technologies like whole exome sequencing and RNA-seq in support of NCI preclinical and clinical studies. So we're basically NCI's genomics lab. And as Kate said, our laboratory is the genomics hub for the National Clinical Laboratory Network. Now, we often encounter clinical trials that plan for the collection and sequencing of archival specimens as part of the biomarker plan. So I thought I would focus my talk this morning on the challenges that we sometimes face for NGS when we're working with archival specimens. All right. And I'm going to start by using the NCI match trial as an illustration of how we think biospecimens should be collected to achieve the best results for NGS. And I would guess that everybody on this call is probably familiar with